All right. So we are going to be talking about the cell cycle, specifically mitosis. A student is examining an onion root tip cell under a microscope. Based on her observation, the student proposes that the onion root tip cell has completed interphase and now it's in the first phase of mitosis. All right. So let's see what happened with this onion root cell. So it's finished interphase and now it's in the first phase of mitosis. The first phase of mitosis is also known as a prophase. 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 Okay. All right. So prophase. All right. So, which of the following pieces of evidence would best support the student's claim? So, in prophase, some characteristics that are important to point out is that the DNA, right, which is your chromatin or chromosome, in this case, it would be chromatin, it would turn condensed, okay? So the DNA would condense, all right? It will condense. Or in other words, it would start foiling up, all right? So the chromatin, which is the DNA by extension, right? Because it contains the the genetic detail, excuse me, uh, it would condense. Now, since it's condensed, it's no longer in its chromatin format, but rather in its chromosome. Chromo chromosome. Chromosome. All right. So now it's in its chromosome format. Okay. And also during prophase, the nucleus, I should say, not nuclear, the nucleus disintegrates. Okay. So the nucleus basically, uh, the nuclear envelope, I should say, it disintegrates, and by extension, the nucleus is gone. Okay. So now that we know the characteristics of prophase, which is the first phase of mitosis, let's look at the options. So the cell's DNA is dispersed within the nucleus as chromatin. This is completely wrong because you can see that it does not introduce any of these criterias, right? The chromatin, which is answer saying is still there, has become chromosome, right? And it's also not within a nucleus format anymore because the nuclear envelope has disintegrated, right? It has disintegrated. All right. So separated chromosome sister chromatids, sorry, are being pulled towards opposite ends of the cell. That's also not happening. This is later on in mitosis. So this over here is during anaphase, a process known as anaphase. All right. So yeah. So anaphase. Sorry. There we go. So that's also not happening because it's much more later on in the cycle of mitosis. The cell's chromosomes are aligned across the metaphase plate. That's the metaphase plate should give it away. Metaphase is actually another process. Uh, it's another, you can say, phase of mitosis, right? Phase of mitosis. So that is also not true. And discrete chromosomes are scattered across the cell's nucleus. This can be true, right? Because you can see that one of the conditions of the chromatin condensing into chromosome is being fulfilled. Now it's also saying throughout the cell's nucleus, which I would believe to be that is probably because it's in the beginning stages of prophase, early stages of prophase. It's later in prometaphase, it's known as, which is later, the later phases of prophase, that the nuclear envelope begins to disintegrate. So that's probably why it's saying the cell's nucleus right there. But this would be the correct answer. Here we go. Okay. The following graph shows how cellular DNA content changes over the course of a typical mitotic cell cycle in rapidly proliferating human cells. So, proliferating, for those who don't know the vocabulary that well, that's completely fine. I get it. It just means increasing rapidly. All right, so just increasing a lot. Okay, so increasing, but emphasized on increasing, like it happens a lot. All right, so we can kind of tell that there's um, a lot of cellular division that's happening, because in order to increase a cell population, you can only do that to through cellular division. I'm just writing CD for sure. Okay, so this graph is telling us how much of the DNA content changes. So our DNA content is just how much DNA there is. So how much DNA is there? All right. The amount, you can say, the amount of DNA. All right. So the amount of DNA, how much is there? Okay. So within here we can see that uh it's telling us the different stages. So G1, S, and G2, these are all part of interphase, and then M stands for mitosis. Okay, so based off this graph, which of the following statements is correct? Human cells spend a majority of the cell cycle within interphase. That is correct. The amount of cellular DNA is halved during interphase. It's actually doubled during interphase. So it's the opposite of this. And we can see that through here, because the DNA content starts at 6, but then during the S phases in interphase, it shoots up, right? It shoots up. And the reason why this happens is because of DNA replication, where within your chromatins in the nucleus, so pretend this is the nucleus of the cell, right? And then you have, let's say, two chromosomes, one from your dad, this is the one from dad, and then this is one from mom. They're in their chromatin format, because remember, uh, throughout the duration of the cell it stays in the chromatin format. So what happens is, during S phase, the chromatins, they actually duplicate, and they create a new strand of DNA. Okay, and then this new strand of DNA is joined together with the chromatin, to form chromatids. Chromatids, right? So it's joined together like that. So now each of the chromatin, instead of only having one DNA strand, has two DNA strands. There's still the same genetic information within the cell, because these new DNA strands that are being created, they're the same thing, right? They're the same exact um, genetic makeup, right? But the amount of DNA that you have, right? The amount of DNA that you have has doubled, because you now have two strands for each of the chromatins rather than only one. Okay. So that is wrong, right? Because it's not being halved; it's actually being doubled. We can see that through the extra strand of DNA that's being added, and that's known as a chromatid. So each one of these strands of DNA are then known as chromatids. And then they in mitosis they're actually pulled apart. So the reason why this even happens is DNA replication is for cell division. So each of the daughter cells have an equal amount of chromosomes in that case, because the chromatin becomes condensed, right? And so yeah, that's the whole reason why this even happens. But these these strands of DNA, as I said, they're known as chromatids. Chromatid, like that. And then chromatin is the whole structure itself. That's the chromatin. This one of these strands is a chromatid. And same thing for the mother DNA. Okay, humans spend the same amount of time in M phase as they do in interphase. That's wrong. We can even see that through the graph. You can see that this is the time, right? You can see most of it is in a large majority of it is within interphase, right? Right. That's all interphase, right? That's all interphase. So you can see that majority of the time is in interphase, and only very, very select few of the time is in mitosis and cytokinesis as well. That's the physical separation. The amount of cellular DNA remains constant throughout the cell cycle. That's also wrong. We just learned that it is doubled, because, and we learned even why and how it's doubled. So that's completely wrong as well. So it's A. Okay. A mouse skin cell containing 40 chromosomes undergoes mitosis. The process results in the formation of two daughter nuclei. So it's going to be two daughters, a nuclei, because remember, during mitosis, what happens is you have your parent cell. So pretend that's your parent cell, and that is separated, or we can say divided, into two daughter cells. So if you have two daughter cells, that means you have two daughter nuclei, nuclei just being the plural for nucleus, because each of these cells contain one nucleus, right? And each of them will contain 40 chromosomes. And the way that you can have 40 chromosomes is actually because of the chromatids. So remember, this is the parent cell, let's say. Let's say this is the parent nucleus, actually, right? Because we don't really care about that the other parts that much. So let me just hmm. Okay, I don't know what was happening. Uh, yeah, sorry, that was some technical issues. Okay, I wasn't able to draw. Anyways, here we go. So we have our nucleus there, and then again, for simplicity's sake, let's say it's only one chromosome, right? Obviously, this is a very simplified organism. Usually, organisms like the mouse skin cell contains 40 chromosomes. But let's say we are have our chromosome here, and it's only one chromosome. All right, you only have one chromosome in the cell. So what happens is during so let's say prophase, so that's the disintegration of the nucleus. So the nucleus no longer exists. So we can actually go ahead and erase the nucleus. So let's say the nucleus has been gone, right? Um oops. What did I do there? Okay. Sorry about that. So the nucleus, let's say it's gone, all right? And so we are left with our singular chromosome, all right? Our singular chromosome, right? Remember, in a real organism like the mouse cell, you would have 40 of these. You had 40 of these, but the same process would happen for each of these chromosomes. So now what happens is during metaphase, it's going to be lined up across the metaphase plate, which is just the middle of the cell. All right. So let's say I'm just going to draw the cellular membrane here. So let's say that's the cellular membrane. So yeah, you're going to have your chromosome in the middle. Then in anaphase, what happens is through the spindle fibers, right? Through the spindle fibers, the spindle fibers are created by the centrioles, which are within the centrosomes. So they kind of create sort of like that type of structure, right? Like that. They connect through microtubules, and those microtubules will push uh, away to the different poles. So poles are at the end of the cells. They're gonna push the chromatids, right? The chromatids, which are each strand of these DNA, to the other sides of the cell, and that it will eventually lead. So this is the same parent cell. It will eventually lead to something like this, right? Something like this, where on each end of the cell you now have each one of these chromatids, right? Each one of these DNA strands is genetically the same thing, um, on each end of these of the cell, right? And each one of these former chromatids are now chromosomes because they're by themselves. They're no longer connected at the point of the centromere. So I didn't talk about this, but they're usually connected at a point known as a centromere. A centromere. So they're no longer connected at such point. They're now by themselves, and then what happens is the nuclear envelope uh, regains itself, and then cytokinesis creates the cleavage furrow, the pinch over here, and then eventually it breaks off the cell into two daughter cells. So, but now you can see that each one of these daughter cells still has one chromosome. It still has one chromosome each, right? Because even though the DNA content has decreased because it's only one DNA strand for each of these cells, it's still two separate chromosomes that were created from that two chromatids. So those two chromatids became two chromosomes themselves. So that's how the mouse cell that contains 40 chromosomes can create actually 80 chromosomes, and then 40 of those 80 chromosomes go to each of those two daughter nuclei because the chromatids are separated, and that doubles the amount of chromosomes, which are then distributed to each of the new daughter cells by pushing them to the opposite poles, and then the nuclear envelope is regenerated again, and then obviously the this cell membrane is broken apart, and then two daughter cells are created, right? Two new daughter cells are created, this being one of them, and then this being another one. All right. Oh, that too is very ugly. Okay. All right. A partial hepatectomy, or a surgical removal of a section of the liver, is often performed to treat, uh, tumors or other abnormal growths in the liver. Following, excuse me, Following a partial hepatectomy, the resting liver cells re-enter the cell cycle, duplicating until the excised portion of the liver has been regenerated. According to the information above, performing a partial hepatectomy causes the liver cells to. All right, so here we're talking about more into the phases, getting more technical with our terms. So, what happens is after the partial hepatectomy, this surgical procedure is done, resting liver cells. So these are resting liver cells, right? Um, Resting liver cells, they re-enter the cell cycle. So what does this re-enter to the cell cycle mean? Well, it's telling us a lot. It tells us that these resting liver cells, I'll just call them RLC. So resting liver cells, they are not part of the cell cycle. These are not part of the cell cycle. Okay, the cell cycle. Instead, they are resting, meaning that they're not part of the cell cycle. Therefore, we have to look when we're looking at our answers, we have to look for a phase that's not part of the cell cycle. Okay, now let's do some review about what is or is not part of the cell cycle. So within the cell cycle, God, okay, within the cell cycle, you have a couple of processes. So within interphase, I'll say I, you have your G1, G2, and you have your G1, S, and then G2 phase. Then mitosis, you have PMAT. That's the acronym that is used to help remember the phases. So prophase, metaphase, anaphase, and then telophase. Ignore that ring. That's just my Pomodoro. And then after that, you have cytokinesis, right? I'm just using C for cytokinesis. I'll just write cyto. Okay, so now that you know these processes that happen within the cell cycle, let's look at some of these phases that might be outside the cell cycle, that might not be mentioned within the cell cycle. And what I see is G0. We don't have G0 as a legitimate phase within the cell cycle. So that means that it has to be outside of this process, outside of this little box, you can think of it. Oops, this little box of where the cell cycles are, right? So it has to be outside of this little box, and that would be the G0 phase, right? The G0 phase. So since the resting liver cells re-enter the cell cycle, they have to start off outside the cell cycle and then re-enter the cell cycle, which it would re-enter with the first phase of the first phase would start off in interphase, right? Because interphase is where the cells first begin, and it would start off in the beginning stages of interphase, which would be G1. So it has to be A because you're exiting the G0 phase, which is outside the cell cycle. I'm just reiterating here. And then you are entering the cell cycle again through the G1 phase because that's the beginning phase. There we go.